Okay, hi everyone. Thank you for welcoming me today to your roundtable. I obviously this is the first time we're here, so I'm gonna have to tell you a little bit about myself and what my lab does, but I'll try to keep that portion really small and jump right into the work that we've been doing on KCNT1. Just to start out, my lab is a translational neuroscience lab. I was trained as a behavioral neuroscientist with Jackie Crawley. She's pretty famous for writing this book, What's Wrong with My Mouse? And so I still get a lot of, do a lot of behavioral work, but these days we do above and beyond behavior. And I've teamed up with a group of people, a group of investigators at UC Davis. One of them you'll hear from in a minute is Dr. Roy Ben Shalom. And then we also work with Dave Siegel, who is pretty famous in gene therapy. He discovered zinc fingers, or he worked with the people that discovered zinc fingers and towels and various gene therapeutic techniques. And Kyle Fink, who's works with Genulta and is considered like a leading expert in induced pluripotent stem cells. And so our goal as a team, we initially were calling ourselves the Center for Interventional Genetics. Then we were told we couldn't call ourselves a center because we didn't have a P50. So we had to go to Program for Interventional Genetics, which means pig over sig. And we didn't like that as much, but that's what we're not going to abbreviate that one. Dave said this quote one time when we were talking to some parents about the fact that we're not the ones trying to find the gene. Someone has already done that. Like, we just need to do something about it. And what we want to do about it is we want to decrease the risk, the time, the money that is involved with starting at drug discovery, as many of you, the brilliant people on this call have talked about, and getting it through preclinical models through to an IND and to get an IND approved so we can go to clinical trials. I think from what I've been hearing, KCNT1 is a lot further ahead than a lot of rare diseases because of the ability to use repurposed compounds that are known to be safe and the fact that so much is known about the target, but at the same time, time is more of the essence for KCNT1 because of the nature of the disorder and the severity of the epilepsy. But I do want to say that the Silverman Lab in general work on a lot of different rare disorders. We're sort of agnostic as to the therapeutic approach that we assess. We work a lot with the Fink Lab and the Nolta Lab, who are sort of CRISPR and AAV gene delivery biased, but we will use repurposed drugs, small molecule drugs, lipid nanoparticles, viruses, hematopoietic stem cell delivery we do in our lab as well. So we're very agnostic. However, whatever approach is working for that particular rare disorder, we will do it. And these are just an example of some of the disease platforms. At the bottom, we've been looking at chromatin modifying genes, gene dose responsive things. So where the gene is missing, you end up with one disorder versus when it's overexpressed, genes at the synapse, and of course, where KCNT1 falls in is at a channelopathy. I don't really have to go through this. It's just that the goal of my group is not just to get to bridge the gap. Yes, we want to get things to patients from the bench as quickly as possible, but we want to do that by not making the same mistakes that were made in Alzheimer's disease research, where we just watched like 600 clinical trials fail. And that was because they were using a lot of information that was based from animal models, but information about neural circuits that were not conserved from the mouse to the human. So we're really big on looking at evolutionary conservation. So we only will focus on functional improvements or improvements in domains that are present from the fly to the mouse to the monkey to the human. So really conserved domains. This was something Tom Insel wanted to do for years at NIMH, and I think it's only really being appreciated now. And we work very closely with the Ben Shalom Lab, who focuses in vitro, and we sort of focus in vivo. And so we are trying to bridge those gaps, and by having corroborating phenotypes in both. We hope that we'll have that. So let me quickly now go through our KCNT1 stuff. So, because I know I only have a minute now. So we worked with Elizabeth Brida and Daniel Davis at University of Missouri, and they actually had made a new mouse model with Bing. Bing, I can't remember his last, Zhao. He made a model that was at the 288 variant, and in humans it's G288S, and in mice it's G269C, but it's at the pore, so it's at the pore domain, and it's a new model, and we have it. And I know that Elizabeth is filing all the paperwork to give it to either MMRC or JAX, but it's definitely available to everyone. Roy's going to talk a little bit about exactly what the mutation does to the action potentials. But we were having trouble with the animals surviving because they're severely affected. And so we use this post-operative care that has been able to help improve the survival from basically 50% survival to almost 80% survival when we're doing EEG surgery. So that's great. We also know that the hets have a phenotype, which has been an issue with some of the other mouse models in this area. And the last pieces of data that I'll show you that are the coolest, I think, is that the heterozygotes alone, when they're given heat as neonates, so mimicking a fever, they actually have they exhibit seizures from that point. So they're pretty severe, much more severe than the wild types. They have them earlier, and the probability that they'll survive post having a fever-induced seizure early in life is lower than the wild types. And so we actually think that this is a really critical phenotype. We saw that they have impairments in some other behavioral assays on their own, just with the channel deletion, but we really want to look at what happens with these early in life seizures and how that advances the disorder throughout life, whether it increases spontaneous recurrent seizures or increases other things. We utilize wireless EEG to do this. They're not tethered or any of these awful, like you usually see EEG look like this, it's tethered. We use it wireless, just implanted in their trapezius muscle, and they live in their home cage, and it's really nice. And from that, we'll be able to see all the different important measures that you're looking at, but that we're talking about with regarding epilepsy, like spike trains and durations, power spectral density, and most importantly, sleep. My lab is really interested in sleep. It's a conserved behavior across species, as well as really relevant for the disorders that we're talking about. And what we do quantity, that's what those EEG telemeters give us, quantity of sleep, but my lab is really interested in quality. And so we're working with a bioengineering student to write algorithms for quality. And I think the last thing I just wanted to show you was that we have the ability to pair those EEG telemeters with whole body plasmography, which tells us about apnea. And if you just give me 1 minute, I'll walk you through this, which shows that this is the activity. So there's no activity. There's an accelerometer in that telemeter that shows you that there's no activity. And then the EMG, you see a little bit of activity. And what we think this is that the head is moving and it's struggling. And these EEG waves, if they were broken down further, you would be able to tell that they're in slow wave sleep. And that's what this big S is for. The program is telling us they're in slow wave sleep. And then up here, what we're looking at is respiratory physiology. So inhale, exhale, inhale, exhale, inhale, exhale. And what you see by the two being paired is that there's a huge apnea, a huge breath hold where they're not breathing and while they're in slow wave sleep, but there's no activity except for these little EMG movements. And so we think that this is actually the visualization of obstructive sleep apnea. And we've seen that to be elevated in rare neurodevelopmental disorders. These different type of apneas you don't have to worry about now. I can explain them on another time. They have to do with whether they're preceded by a sigh or followed by a sigh. But the important thing is that we are able to detect this, and we think this is going to be a great either biomarker or metric for therapeutic delivery. And we were able to put these brains in MRI and see some subtle differences. Right now, what we're doing is processing brains through for MRI that have been through those early life fever-induced seizures. And so we'll have all that data for you as well on our next call. And that's about it. This just says what I said already, that we're really agnostic about the type of therapeutic, but that we're working with at least those 1, 2, 3, 4, 5, 6 types. And I'll answer any questions that you have.
Thanks, Jill. Fantastic work. If anyone's got a question, let's do one or two now.
Can I ask if the patients, these children who have the channelopathy, are they treated with a CPAP or some type of device during the sleep periods? Is that possible to prevent this sudden death syndrome that you see in these patients?
I can answer that. I'll say lots of patients are treated with CPAP overnight. Most patients do have some form of oxygen at home, and the parents sometimes put them on something overnight. Whether or not they're on CPAP just kind of depends on whether they've had a sleep study showing sleep apnea. But I'd say it is quite common for kids to be on CPAP or BiPAP at night.
Does that help the mortality?
We don't know. We don't really have enough information about that. And it is also a little bit probably multifactorial because if you're on CPAP or BiPAP at night, you almost certainly have an oxygen saturation monitor on. And so there's a correlation, between that and being less likely to die during sleep. Yeah, I think it's something that we need to collect more granular data about cause of death in the KCNT1 population. And that's something that folks from the foundation are definitely interested in doing. I think one of the challenges is just that it's such a rare disease in such small numbers that it's hard to get the numbers necessary to really assess cause of death extremely accurately, but we do definitely want to get better at trying to understand that.
Yeah, I think there must be a big central apnea effect.
Right.
I
So
would say.
that's actually what I was gonna say. That our lab came into sleep apnea by on the side. It wasn't that we meant to study apnea. What we wanted to do was to have the respiratory information. We actually just tried to apply for a grant to also get the cardiac physiology all integrated with the neurophysiology at the same exact time, so that we could see that any model that died uh overnight or died from a spontaneous recurring seizure, that we would be able to capture the respiratory effects that were going on there. And then we wanted to distinguish central apnea from obstructive apnea, because in a lot of other rare disorders where there's facial dysmorphia, there's a lot of abnormal breathing patterns, either hyperventilative breathing patterns or obstructive breathing patterns. So we needed to be able to dive into that more clearly and really know what's going on. And that was our purpose in doing it.
Sarah just put in the chat that the foundation is currently working on developing a protocol to better understand mortality in KCNT1. And I think that is really important. We do encourage all families to get evaluated for sleep dysfunction like obstructive sleep apnea or central sleep apnea. But I think one of the things that would be useful to understand from looking at mortality would be are simple interventions like just putting kids on CPAP or BiPAP and having an O2 sat monitor. Is that potentially associated with decreased rates of mortality? I think we see kind of two trends in mortality overall, just anecdotally. One is that in the younger kids, especially the babies, what sometimes happens is that the seizure burden just gets to be too much, and they basically can't be kind of at some point the team decides that the amount of kind of sedation that's required is just not really sustainable, and then there's withdrawal of care. And then the second thing is, kind of this SUDEP type picture where kids are just, basically dying in their sleep. And we don't know whether that is because of, apnea, and if so, if the apnea is related to seizures or not. And so I think, that's something that would be really important for us to try to figure out as part of this study that's going to be going on. And so I'm grateful to you for looking into this in the mouse model, because I think that's really important work.
All right, Sarah, go ahead and ask your question, then we'll move on.
Yeah, okay. Thanks, everyone. And thank you, Dr. Silverman, for your presentation. I just wanted to weigh in on kind of the dysmorphology component from my perspective as a genetic counselor. So we don't really see true dysmorphisms or facial dysmorphisms that we expect in KCNT1 that we expect to contribute to any type of like obstructive breathing patterns. And I just wanted to second kind of from what we know about mortality in KCNT1, most of it is, if there is an apnea-related death in general, we know that it's tied to, or we think that it's tied to seizure most of the time. So I just wanted to weigh in from kind of the clinical side a little bit.
That's great to know. Maybe we could talk more offline about that, because we really need that information to sort of design the experiments properly.